landscape mapping, how it was discovered through macromolecular connectivity landscape mapping of the mammalian brain. So could you just confirm that you can see my slides?
Yes, we can.
Super. Let's try and make this big. So I'm going to put the acknowledgement slide up front because it took such a huge number of very talented individuals to get this work published. The proteomic and machine learning work was done in the laboratory of Andrew Amelie at the Center for Network Systems Biology at Boston University. And the functional and molecular work was done by me as a postdoc in the Department of Pharmacology under the supervision of Benjamin Wollerson. But there's been some other really useful collaborators who've provided reagents and critical experiments, and thank you to all of them. So this work was published at the start of this year in Cell Systems under the title Brain Map Elucidates the Macromolecular Connectivity Landscape of the Mammalian Brain. And the resource is live at the website that you can see at the bottom. And I invite you all as neuroscientists to explore that resource, to look for the interactions, the protein-protein interactions of your favorite gene of interest. So how was brain map generated? Well, the brain interaction map is made by taking mouse brain and lysing in non-denaturing conditions to preserve the integrity of native protein complexes, and then performing tandem orthogonal fractionation using a series of different biochemical fractionation methods to isolate protein complexes based on different chemical and physical properties. These are fractionated using HPLC, and then the fractions, the proteins in the fractions are identified by quantitative mass spectrometry. So this is the flow through of the protocol. And in B, you can see all of the experiments, all of the fractions from the 9 different separation methods. Each column represents a fraction, and each row represents a protein with the heat map representing the intensity of the signal of that protein within that fraction. And then what you do is you use machine learning to identify native endogenous protein complexes based on the principle that proteins that reproducibly co-fractionate together do so because they're part of stable macromolecular complexes. So this would be this work up here. And what you do is you teach your algorithm based on a series of well-characterized known complexes, and then apply that algorithm to half your data set as a training cohort, and then benchmark against the second half of the data set later. And what does that look like? Well, here is a broad survey of the neuronal complexes on the mouse brain. And each of these little spots represents a complex identified. The majority of them are annotated, but we believe that we've added depth and new annotations, new subunits to those previously identified complexes. And then a bunch are novel. And in each of these complexes, you can see there's little blue spots, and those represent proteins that have neuronal annotations, which you would expect if you did this sort of experiment on mouse brain tissue. The other thing that you'd expect is that a lot of the components of these complexes have strong links to a diverse set of human neurological disorders. Again, schematically, you can see all of the complexes represented as little spots here. And this time, all of the purple quadrants represent the protein components that have either a genetic or pathological association with some neurological disease. And you can break this down based on the various protein aberrations in a number of developmental and neurological disorders. If you look at the association of these variants, you find that they are highly associated with diseases of the nervous system, and less so with diseases that are not associated with the nervous system, which is excellent. That's what you'd expect. So what do some of these complexes look like? And I think I've got five complexes here. There's just an overview of some of them. So complex 35 contains alpha, beta, and gamma subunits of the G protein coupled receptor. So these are broadly expressed in both neurons and non-neurons, and are really, really important for cell signaling pathways. Complex 20 complex 20 is composed of a number of cell adhesion and cell signaling systems between axons and the myelin sheath, and so is probably involved in trophic support of axons by the myelin sheath. Complex 65 here contains a number of vacuolar protein sorting associated proteins, VPSs, and sorting nexins, and is probably involved in endosomal delivery. The complex is involved in targeting vesicles for endosomal delivery. Complex 42 contains a number of SNAPs and other interesting things that are involved in the snare complex. So this is a really important complex for vesicular trafficking and vesicular fusion as part of neurotransmission. And then complex 251 here contains the neuronal specific neurofilaments, medium, heavy, and light. And you can go back and you can annotate the subunits of these complexes for their association with neurological disorders. So for example, in complex 251, you have these neurofilaments that are associated with ALS. And in complex 20, you have the prion protein, which obviously is involved in transmissible neurodegeneration, but is also associated with myelin sheath maintenance, which is why it's part of this complex. So we wanted to identify gene ontology and molecular function of the complexes. And so we applied the gene ontology analysis to complexes, and we find a number that have a high association with RNA metabolism and RNA binding. And that is something that we're really excited in. And so we focused on RNA binding protein complexes, which you can see here, the complex numbers and the molecular function terms associated with those complexes. And there's one in particular that we're really excited about because it contains a number of components that are genetically and pathologically associated with ALS. So here's TDP43, here's FUS-TLS, here's ataxin-2 and TA1. TDP43 is the major pathological protein of ALS, and those other 4 proteins are associated with either the pathology or the genetics of familial ALS. So we validated the brain map work by reciprocal immunoprecipitation of either TDP43 or another component of the complex, HNRMP-H1, from wild-type murine brain. If you pull down TDP43, you find association with HNRMP-H1, DDX5, TA1, and FUS. And similarly, if you pull down HNRMP-H1, you find other components of the complex. So why is TDP important? Well, TDP mutations are causative of amyotrophic lateral sclerosis, or ALS. TDP43 is also the major component of ubiquitinated inclusions in frontotemporal dementia and ALS dementia, ALS, including in sporadic ALS. So you can see here in an affected neuron, TDP43, which is supposed to be diffusely nuclear, redistributes out of the nucleus and into the cytoplasmic inclusions. So that's the TDP pathology in ALS cases. One of the other interesting components of the complex that we discovered was ataxin-2. So in ataxin-2, large polyglutamine expansions are responsible for generating spinocerebellar ataxia. But intermediate length polyglutamine expansions are associated with an increased risk of ALS. And one of the collaborators on this work, Aaron Gittler, has actually previously shown that therapeutic reduction of ataxin-2 extends the lifespan and reduces pathology in TDP43 transgenic mice. So here are some cultured U2OS cells. And Lindsay and Aaron have knocked down ataxin-2 and show that in these cells, you get a reduction in TDP43 inclusions. If you take TDP43 transgenic mice, they die very quickly. But if you knock down ataxin-2 either as a heterozygous or a homozygous, you get increased lifespan and better behavioral and motor outcomes. And so what we wanted to do was explore how the complex of TDP and these other RNA binding proteins changes in a disease state. And we did this by doing TDP43 immunoprecipitation from either TDP transgenic mice or TDP transgenic mice that are protected by the depletion of ataxin 2. And then this is a volcano plot of proteins associated with TDP43 detected by quantitative mass spec. Anything on the left shows reduced interaction with TDP43 in the protected ataxin 2 knockout mice or knockdown mice. Anything on the right shows increased association with TDP43 in the ataxin 2 protected mice. And I've highlighted here all of the detected members of the complex 22 RNA binding protein complex. So first of all, this validates the brain map co-association platform. The other thing that's really interesting is that all of these proteins show reduced interaction with the pathogenic TDP43 in the ataxin 2 depleted mice, with the exception of DDX1, but that's a net negative change. The ones that are highlighted in red are those that are statistically significant, and we were able to validate a couple of these H&RP-H by IP of TDP followed by Western blot as well. So the TDP pathology in those TDP transgenic mice also disrupts the distribution of RBPs in that complex. So here we're performing fluorescent immunohistochemistry of the transgenic TDP43 in the transgenic mice and in the wild type mice. And you can see that in affected neurons, you get redistribution of human TDP into the cytoplasm. And in instances where this happened, it also drags out H&RP-H1, DDX1, DDX5, and ILF3, which are components of that complex. And in some instances, it also forms these inclusions. And you do not see this in non-transgenic animals. There's no redistribution of their RBPs. So we also wanted to look more deeply at the changes in the proteins that are associated with TDP43 in the transgenic mice and in the protected mice. So we performed gene ontology molecular function analysis on proteins that show decreased interaction with TDP43 in the protected ataxin 2 brain. And a lot of these are either nucleotide or RNA binding proteins. And those proteins that show increased interaction with the transgenic TDP in the protected ataxin 2 brains are those that are involved with cytoskeleton, with binding kinases, or parts of the unfolded protein response. And so I've labeled classes of proteins in the volcano plots, as you can see here. It's a bit busy, but generally you can see that a lot of the proteins that are annotated as RNA binding with RNA binding functional terms, such as the H&RPs, show decreased interaction with TDP43 in the ataxin knockdown protected brains. Whereas here, these are functional categories associated with protein folding and ATP binding. Here are some heat shock proteins, for example, and the majority of these show increased interaction with the pathogenic TDP43 in the protected brains. The other thing we wanted to do with BrainMap was try to identify regional distribution of complexes. So we dissected mouse brain in the regions that you can see here and performed total proteomic analysis of expression patterns of proteins. This data is available on the portal that I showed the link to earlier. I'm just going to focus on the complex 22. And you can see here all of the expression distribution of each of the proteins of complex 22 in the regions that we analyzed. What's really surprising is that complex 22, the proteins show highest expression in the cerebellum, which is an area that is unaffected in ALS. And in the area that's mostly affected in the spinal cord, in ALS, the spinal cord and the frontal cortex, there's actually not that much expression. So that was a little surprising. But if you look at the regional expression of TDP43, and this is a protein that is very well associated with the pathology of ALS, it shows a really nice correlation. So TDP43 is the blue line here. And it shows really nice correlation with the summed expression pattern of complex 22, which is the dotted red line. And you can see again that TDP shows high expression in the cerebellum and low expression in the spinal cord. So So expression of TDP43 or the complex alone doesn't necessarily define which region is susceptible to ALS. It may be that this is a function of the cell types that you see within these regions, and we're going to do some more work to try and figure out if this can tell us anything about why the spinal cord neurons are so susceptible to TDP and hopefully complex 22. But yeah, so brain map contains a lot of this data, regional and cell type specific distribution of complexes. So here's a complex, for example, and it shows you regional and cell type specific distribution. And I invite you to explore all this data through the portal again, which I'm really going to sell. The last part of this work was to look at functional aspects of complex 22. And we did this by taking SH-SY5Y cells and knocking down components of the complex. Here, TDP, H9P-H1, and DDX5, and then looking for known functional outcomes from that. And there's one really well-assayed function of TDP, and that is as a regulator of alternative splicing of exon 17B of sortilin-1. So TDP43 actually binds upstream of exon 17B, and it's been previously shown that when you deplete TDP43, you get aberrant inclusion of exon 17B. That leads to a premature stop codon, nonsense-mediated decay of sortilin-1, and that actually has some consequences on the ability of sortilin-1 to interact with progranulin, which is also a gene which is genetically associated with frontotemporal dementia. So this data was known already. If you knock down TDP, you get aberrant inclusion of exon 17B. If you knock down DDX5, you also see a trend towards inclusion of exon 17B, but this wasn't significant. If you knock down the two together, you get very dramatic inclusion of exon 17B, and this is higher than either of the single knockdowns by themselves. If you knock down H9P-H1, there's no real effect on inclusion or exclusion of exon 17B. But what's really neat is that if you do H9P-H1 knockdown at the same time as TDP and DDX5 knockdown, the absence of H9P-H1 blocks the effect on inclusion of exon 17B brought about by the knockdown of TDP43 and DDX5. Emmanuel Bratti has done some really nice work showing that these proteins all have a binding site in a splice regulator upstream of exon 17B. So we know they all bind in the RNA upstream of 17B. We know from our work that they interact together, and now we can show that they have a function. So we believe that H9P-H1 regulates the exclusion of exon 17B, and that DDX5 and TDP43 are negative regulators of that. So this is a neat discovery where we've shown that there's an RBP with that has a functional module in which members exert antagonistic effects, right? So some increase inclusion, some increase exclusion, and the interplay between them is what regulates the final output. So just to conclude this part of the talk, we defined a global interactome of multi-protein complexes using cofractionation mass spectrometry. BrainMap shows assemblies that show distinct functional, physical, regional, and cell type specific attributes. Many of these are linked to neurological disorders, and we have identified an RNA binding protein complex that's associated with ALS, which we validated, we showed was responsive to disease state, and we have shown that it acts as a functional module in which members exert antithetical effects on exon usage. So the BrainMap is a really neat neuroscience resource for mechanistic exploration of protein complexes. And again, I remind you of the portal. And so I think I have a little time left. So I'm going to, oh, here's the portal. Yeah. So you can see if you search for your protein of interest, your TDP, it'll tell you some details about the protein itself, those things that are interacting with it, and then it gives you links to the complexes that you can explore. So what's next with this work? Well, we're really excited to apply BrainMap to disease tissue to see whether or not there are changes in complex composition and function during disease. We're going to try doing this with AD tissue and ALS tissue, and we can also apply this to transgenic mice, but reviewers seem to be less interested in that than the human tissue. And then in terms of looking at the RBP itself, the RBP complex itself, we're going to do a much deeper investigation of how the complexes are affected in ALS-affected tissue. And then the thing that I'm really excited about is looking at how complex components change as disease progresses. And to do that, we're going to imply split proximity labeling of the RBP complexes. And this should allow us to interrogate complex interactome changes in basal and disease states. So how does that work? Well, TurboID is a proximity labeling protein. And then in the presence of biotin, it biotinylates proteins that are nearby the TurboID. And if you fuse TurboID to TDP43, you get a different profile. You get the things that are proximal to TDP43 at the time of addition of biotin. And then you can split TurboID. So this is work by Alice Ting. She's very kindly shared the constructs with us. You can split the TurboID into an N-terminal and a C-terminal region, which by themselves don't really have any effect until you fuse them to TDP43. When you fuse them to TDP43, they have to wait until TDP dimerizes for split complementation of the TurboID portions. And then in the presence of biotin, you get a different pattern of biotinylation versus the TurboID, the full TurboID fused to TDP. So what does this tell you? This tells you a protein interactome that's nearby TDP43 when it is dimerizing or arguably aggregating. So this is work with TDP43, but to validate the RBPs multimerize, we've also used split Venus. So here we have an N-terminal and a C-terminal of split Venus fused to either TDP43, HNRMP, H1, and DDX5. And you can see that they are often able to split complement homogeneously. So this is DDX5 and DDX5. You also get really nice heterogeneous complementation. Some cases you get it better than with the homogeneous complementation like this. So just for the last part of the talk, this is some unpublished data that looks at how an RNA binding protein called TIA1 potentiates phase separation of microtubule associated protein tau and promotes the generation of toxic oligomeric tau. And the reason this is important is because it looks at heterogeneous interaction of RNA binding proteins with the disease protein and how that affects the chemical and biophysical properties of either protein. So phase separation is a hot topic. It's a really neat phenomena where you have a protein with an intrinsically disordered conformation that under the right conditions, usually the addition of crowding agents, undergoes phase separation to produce droplets of liquid protein in vitro. And tau does this. So here is green labeled tau that in the presence of RNA and increasing concentrations of polyethylene glycol forms droplets of tau. But polyethylene glycol is not a very physiological component. So we were looking for means by which this might occur physiologically. So again, what we've got here is green labeled tau. And in the presence of RNA and increasing concentrations of recombinant TIA, you can get tau to phase separate in the presence of the TIA. And this is quantified on the side here. TIA1 also facilitates biochemical changes in tau. So if you do fluorescent recovery after photobleaching on droplets of tau, they very rapidly recover after the area that's been photobleached. But if you add TIA1 in increasing concentrations, you reduce the recovery of the area that's been photobleached, suggesting that tau becomes vitrified, gelate in the presence of TIA1. If you look microscopically, if you look at the changes in the distribution of tau, here we have 5 micromolar tau. These are always labeled in green. And then increasing concentrations of TIA1, in this case, labeled in red. When you have low concentrations of tau and TIA, both the tau and the TIA are diffuse within the droplets. But as you increase the concentration of TIA, you redistribute tau into these concentrated microdomains. We've quantified this in a way you can see here. And it turns out that the partitioning behavior that you see of tau is dependent on the RNA binding protein that you use. So here we are using 5 micromolar of tau, again in green, and 5 micromolar of the RBPs that are listed here. And at the high concentration here, you can see that there's different patterns of tau. You either get microdomains of tau within the RBP, or you get microdomains of the RBP within the tau, or sometimes you get no mixing of the two proteins. And we believe that this is a function of the energetics of the heterogeneous and the homogeneous interactions. You have to have favorable energetics, heterogeneous energetics for the two proteins to co-phase separate. But the strength of the homogeneous interactions define whether or not one protein will form concentrated microdomains inside another, or if it's the other way around. And so just to conclude this little part, we just want to highlight that TIA1 can drive phase separation of tau at physiological concentrations without the need for an artificial crowding agent. And that the results on the distribution of droplets suggests an organizing principle for coordinated phase separation, where you can bring together functionally relevant proteins to promote a particular biological activity in a membraneless organelle. And this is something that we would like to explore with the ALS complex 22. That's really the end of my talk. I just wanted to, if there's time, one of the other aspects of that latter part of the talk is that we look at the relative toxicities of the changes that occur in tau. So we've known that RNA and other polyanionic compounds drive fibrillization of tau. But if you add TIA1, you can actually inhibit the formation of fibrils and lead to production of oligomers. So I'm just going to highlight panel D here. If you add RNA and TIA, you get a large amount of TOC1 positive oligomers by ELISA. This is a TNT1 antibody that looks at a conformational change in tau. So these are fibrillized in the presence of RNA, but then oligomerized in the presence of RNA and TIA, and you get much more oligomers. And if you add polyethylene glycol, under no conditions do you ever get oligomerization of the tau. So this doesn't appear to be a very physiological aspect. And then finally, we applied different fractions to cultured neurons, either tau incubated in the presence of polyethylene glycol, or tau fibrillized in the presence of RNA, or oligomerized in the presence of RNA and TIA1. We find that the oligomeric tau is the most toxic, leading to the greatest release of LDH and the highest death of neurons when they're imaged by microscopy. And so we have this model where addition of RNA leads to oligomerization of tau, which will convert to fibrils. But in the presence of TIA1, you inhibit the conversion of oligomers into fibrils and leave them as oligomeric tau that is highly neurotoxic. And with time, we believe will also form phase-separated and gelated droplets. Thank you very much for listening. I'd be happy to answer any questions you have, and thank you.